everybody as you're joining from the waiting room. So we're very pleased today to have another webinar from ATP-Bio, and we're going to be featuring our trainees today with a presentation from Leo Liu, who just graduated, I understand, from UC Berkeley in Boris's lab and is just starting his postdoc in UCSF. Who will you be working with in UCSF, Leo?
Christopher Hernandez. I will work in the engineering material for my postdoc.
Okay, is it a step to the side or is it a continuation of some of the work you've been doing with Boris?
Kind of like broaden my research, like it's something different, but I can still use my previous like experiments.
Excellent.
Hmm.
So Leo is going to be talking to us about his work in cryo printing during his PhD, which will be very interesting. And then that'll be followed up by updates from the International Cryobiology Young Researchers Group from Dr. Lakshya Gangwar, and then also an update from our Scholars Leadership Council from Alexander Ulate. So welcome again to everybody. And I think without further ado, Leo, if you're ready, we'll just
Sure.
pass it over to you.
Let me share my screen. Can you see my screen?
I do.
Yeah, great. Hi, I'm Liu Lu. I'm a PhD just graduated from Dr. Boris Rubinsky's lab from UC Berkeley. Today, I will present our work for temperature control cryoprinting for food. Food is one of the most important components of human life. It is not only sustains us by providing the nutrition and energy we need every day, but also shapes our health, culture, and the society. Yet, food can also become a major problem when access, quality, or safety is lacking. According to the data from the Office of Disease Prevention and Health Promotion, 60% of the US adults with chronic disease. 18 million people experience food insecurity. In the meantime, the US investment in the healthcare is over $4 trillion in 2020. To address these upstream causes of poor health outcomes, the conception of food as medicine can be a potential solution. Such conception of food as medicine can be traced back to 400 before Common Era. A Greek physician, Hippocrates, proposed the conception, which lets the food be the medicine and the medicine be the food. Many other cultures also share the similar conceptions of treating food as a form of medicine. One important emphasis for food as medicine is personalized food. Such kind of emphasis of the personalized food is becoming more and more practical in today's society with the rise of new technology like advanced manufacturing, for example, like 3D printing for food. We now have the ability to customize food in ways that were not possible before. Such technology allows us to precisely control the shape, the structure, and even the nutrition of the food we make. As a 3D printing process will print food in voxel by voxel, it is possible to control the food from the voxel level. In a typical extrusion-based 3D food printing process, there are two key steps: extrusion and deposition. First, food inks are pushed through a nozzle and then they are deposited layer by layer, following a computer-aided design. I like to highlight a few important milestones in 3D food printing. The very first 3D food printer was developed back in 2006. In the early days, the technology was quite limited. It could only handle simple foods like chocolate and cheese. As the field advanced, researchers began exploring more complex possibilities, such as printing meat-like foods with customized nutrition and texture. More recently, some approaches have even moved towards embedding living cells directly into printed foods. However, The field is still missing a breakthrough technology, a real killer app that can overcome some of the major challenges in food printing and make the products truly attractive. For example, current research has shown that 3D printing can create food that looks good, but the bigger challenge is how do we print food that not only looks appealing, but also mimics real food in both its macro and microstructure, instead of just replicating its appearance. To overcome the challenges, we introduced the temperature control cryo printing for food application. While the technology is originally designed for the biological tissue fabrication, food actually can also be treated as an inactive tissues, which means food printing shares many similarities as a bioprinting. The biggest difference is like we don't care about whether food is live or not. The biggest difference between the TCC, the temperature control cryo printing, and the traditional printing is the printing materials gets frozen, and during the frozen process, the ice generated can support the printing structure and modified microstructure. Because the ice crystal can be very tiny, can be very small, like down to several microns. So it can be help to create such kind of similar level of the microstructure of the printed structure. In In a practical way, there are four main factors that control ice crystal formation process. These factors are temperature of the printing ink, the temperature of the printing bus, the height, and the height between the printing nozzle and the printing layer. By controlling these factors, we can control the freezing rate, which affects the size of ice crystals. The size of ice crystals can then change the microstructure of the printed sample, which can be as small as 10 microns. This process can potentially improve the resolution of the traditional printing by creating more finer microstructure. So my presentation today will be divided into three parts. Start with the texture. The first thing we focus is the meat, since it has some unique features like fibrous structure and the improvement cuts, the marbling texture that makes it especially valuable. So the question here is how to create such kind of like interesting features. Start with the microstructure. While the regular freezing like can modify the microstructure, as you can see here, like samples before and after freezing, which after freezing it shares like more porous like structure, but it cannot produce a fiber-like microstructure, just like the real beef. So here we get inspired by nature. Like in the nature, there is actually kind of needle-like ice crystal. So can we transplant this kind of mechanism in our printing process? The answer is yes. So we introduce directional solidification. So directional solidification is a technology which moving the samples from a temperature-controlled hot plate to a temperature-controlled cold plate. Here we have four parameters: the temperature of the hot plate, the temperature of cold plate, velocity for the samples moving from hot plate to cold plate, and the depth, the length between the hot plate and cold plate. By controlling these four parameters, it is possible to control cooling rate and cooling direction. So, which finally will help us to generate such kind of needle-like ice structure. And this kind of needle-like ice structure can help to create fiber-like microstructure. So as you can see here, like the samples from three different conditions. The first one without freezing, the second one with a no uniform freezing, and the third one with directional solidification. And this can show how to potentially the freezing, a controlled freezing method can help us to create fiber-like structure. So marbling is one of the defining characters of the meat, especially in pre-dip cut. Traditional 3D printing, 3D food printing try to recreate marbling by using multiple nozzles to print different inks. The issue is the marbling usually ends up looking broken and disconnected, so the final product just isn't that convincing. So what we did instead was to develop a coaxial printing strategy. This approach lets the materials mix more smoothly, and as a result, we can create marbling patterns that looks much more natural. The next part is binding, which is designed for the scalable manufacturing. So to extend the technology to broader applications, it is necessary to scale up TCC for like industrial level manufacturing. The challenge is that the regular 3D printing process doesn't really fit into an industrial production line. To address this, we broke down the TCC process into modulars, each step handled by its own machine. Under this situation, the 3D printing becomes 2D printing. Each layer will be printed separately. They get frozen separately and be assembled into a 3D structure in a frozen state. So this actually raises a key question here is, how do we bind these separate 2D printed pieces together in a frozen state and finally have a 3D structure? This video also show like the 2D printing process for meat. So I want to take a broad view here. It can also, because such kind of technology can be also applied to a bio tissues. While this binding approach can certainly be used for the food, just imagine building a complex multi-level tissues which each piece has its own unique properties. Instead of trying to make everything in one general process, an alternative way is to fabricate each piece individually and then assemble them together, just like building with Lego. So we developed a new technology called cryo here. So idea here is pretty simple. When we have two frozen printed pieces, we can join them together by applying a special cryo ink at the interface. This ink melts just at the contact region without any external heating. By adjusting the chemical potential, a familiar example is how salt melts snow on the road in the winter. Once the interface melts, the materials can diffuse and interact across the boundary. Afterward, we refreeze the assembled structure at a lower temperature and alter and let it fully salt and cross-link. What's exciting is that like the final cryo sample shows no significant difference in the tensile strength compared to the samples that were original printed as one piece, just as shown in the in the bottom right. The choice of the cryo is also flexible. For example, like for food applications, we can use food-safe salts to make the process compatible. So the binding technology expands the complexity of the structures we can print with TCC, like heterogeneous directional structures. The letter C here is assembled by 3 printed lines with directional structure, while the technology can also be used to help to fabricate base-free complex, which is challengeable for the 3D printing, traditional 3D printing, as the upper layer usually is relied on the bottom layers in the 3D printing process. So finally, I would like to talk about one case. We use our printing technology, the dysphagia. So dysphagia is a condition where people have difficulty swallowing, making it hard for them to eat regular food safely and comfortably. The food served for dysphagia need to specially modified. We are collaborating with the USDA on the project compared with conventional 3D food printing. Temperature-controlled cryo printing has a potential to create more tailored features for dysphagia care by modifying food texture. Similar to the approach described in part 1, our work can be divided into 2 complementary directions, the microstructure design and the macrostructure design, just like the part 1 in the presentation. For instance, TCC creates directional structures that give food an isotropic shape properties, similar to the chewing experience of the meat with fiber texture. In the top images, you can see how adjusting the freezing rate changes the size of the fiber-like structures, which affects their shear performance. While not shown here, our data also suggests that TCC printed food can vary hardness and adhesiveness. Such kind of texture profile analyzer data for the food without changing their actual composition. We tried both vegetarian and non-vegetarian inks to print food for people with dysphagia, and all of them passed the IDDSR test. The test is the standard way to check if food is safe to swallow for dysphagia patients. We hope this work can be further help the dysphagia food preparation. Thank you so much for your listening.
Great. Thank you, Leo. That's very interesting. Oh, I see you've got this is from
Yeah,
our UC Berkeley annual meeting, right?
yeah.
Very nice. Okay.
So,
So we'll open it up for questions. I guess I had one to start. In terms of the dysphagia and the folks that you're addressing, are you working with clinicians or is this with the USDA? Can you give us a little more background in this really exciting and interesting application
dysphagia.
area? Yeah,
Yeah.
like, I understand that you can produce this and then
Mm,
and it's a collaboration with the USDA. How
mm.
does this ultimately get to patients? Do doctors have to get involved, or is this a selection that someone would just make on their own?
So, for our technology, like apply for the dysphagia, like right now we are in kind of like a pretty early stage. Like we show the technology and we test with some food inks and print some dysphagia friendly food, and we run some like standard test, like the IDDSI, the texture kind of stuff, and we definitely are very interesting to apply this like this food like serve this food for dysphagia, but like this process can be a little bit like more complex because we need to get approved like by the IRB kind of stuff, and I think for the USDA part, like they are still looking for some potential partnership here, and yeah, so that's general.
So, okay, and maybe
Mm,
just a quick follow on, but has
mm, mm.
this have you demonstrated your like within a cohort of people that have dysphagia that this helps them?
We actually haven't tested with a dice feature. Like so like right now our standard is utilize some like uh standard test like IDSI, because if we want to test the food with real dice feature patient, it needs like kind of like a super complex paperwork, and right now like we are still waiting for the approval for that. Before get approval for that, like it we cannot like do that.
Yeah, that's fine.
Yeah,
I'm just
yeah.
asking.
Mm, mm,
Great. Let's open it up to others
mm.
for this interesting talk from Leo. Are there any other questions? I can't actually see. Let me look and see if anybody wants to raise their hand or, okay, I don't see hands being raised. So I'm going to keep going for a moment, Leo.
Thank
Um,
you. I
yeah,
will stop my
so
share.
as you go to UCSF and you're working in your postdoc, are you going to continue in the cryoprinting field or are you moving into a different field?
It's kind of like a different field, but the point I think why like my peer here I'm is I can potentially utilize the use the cryo printing to for their application. So the big field I work here is engineered living materials. So which means like material with live bacteria. So make it like kind of functional materials. So I think uh a potential um project here can be like how to apply, how to utilize the TCC to fabricate like such kind of materials, and how can we, for example, like try to preserve it if like the material is really like interesting, have some like specific function, and that's something I think I'm very interested for my future postdoctoral work. Yeah.
Great. All right. Thank you, Leo. Last call out there. If there's anybody who has a
Hey
question.
Leo, I was wondering after you 3D bioprint these, is there like a
Mm.
polymerization step or do you have to like polymerize it before you cryopreserve it?
Sorry, could you repeat?
Do you have to like polymerize your meat
Oh yeah.
together or something along those lines?
So yeah, so there is a polymerization process, like step, which is actually at the like the final step, because after you TCC printed is in a frozen state, and if you want to use the product, you definitely need to like soy it, and like the polymerization process or like cross-linking process step here is actually together with the sawing. So doing a sawing process, like the whole structure will be cross-linked, and they like including the binding. That's actually the cross-linking is the mechanism which finally get two pieces together. But yeah.
Have you tried to freeze it before or after cross-linking it?
So if we freeze after cross-linking, the microstructure can well be like less get influenced because cross-linking is pretty strong. It will contribute, like it will like make the mechanical property pretty strong, and the ice crystal may have less like influence towards the like final structure compared with the samples we freeze first and cross-link later. Yeah, so the cross-linking, you can also like treat it as like kind of a fixed step, which you can try to make the structure remember how the structure it is during the freezing process.
I had a question. Sorry.
Yeah.
Go ahead, Catherine. Thank you.
Yeah. I had a quick question too. Some of these food you prepare with meat, right?
Yeah.
Is there any plan? I mean, are you gonna eat? Is this food supposed to then be eaten cold, or will there be like a heating process? Okay, will you be heating this for the real patients, or is there a danger of eating it raw?
So right now, all the food we print here are ready to eat because we treat the materials before, like we already cook the material before the like the printing process. But that's actually a very good question. It's like how we finally like treat the printed stuff, because like cooking, it's a kind of complex process. It definitely will influence structure a lot. So like we haven't tested like how the like material, how our printed structures changing during the cooking process. Here's like this can be, I mean, depends on how you cook it, how the temperature. There are so many like factors here. To make it like more practical, like right now, what we do is we just pre-cook the material, or like some material, right? We used here ready to eat, and we can reheat the printed scaffold. I think like reheat is fine, but cooking can be something like right now we are not sure. Yeah.
Okay.
Great. So Leo, thank you very much for that talk. If you could please send me that photo. I don't think I have that nice photo of the group of folks that
Sure.
worked with Boris over the years. That's a very nice photo. And I also want to take this opportunity to welcome our ATP-Bio partners that I see on the call, Calvin and also Raphael from MightyGen, and I think Nathan Allen. And if I missed anybody, I'm sorry about that, but thank you all for joining us. And I think now we'll transition into ICYR from Lakshya, and I'll also identify Charles Elder, who is our Crystal Award winner from Cryobiology this year. Charles, so thanks for joining. All right, Lakshya, over to you.
Hey, thank you. So I do have a couple of slides, if I can share them. Let's see. Let me know if you can see my slideshow.
Yes.
Okay. All right. So thank you for the stage. And I am Lakshya. I am a postdoc in UMN in Dr. John Bishop's lab. I've been part of ATP-Bio since its inaugural time, 2020. So I've been. Hello?
I think you're probably okay.
Yeah.
I'm in.
Yes. Emily, I think you might want to.
Yeah, she's muted now.
Okay. All right. Yeah, so and I've been part of SLC in past, and that sort of ignited my interest to go beyond ATP-Bio and see what young people in cryobiology are doing. That's where International Cryobiology Young Researcher Organization comes, which is essentially a early career wing of Society for Cryobiology. And I'll just give you a quick update of what it has become. It has been there for a really long time, since the society was founded, but more recently, after pandemic, we've tried to make it into this virtual Zoom era. The mission is in the name. We just want to build a community of people who are around the world who are interested in cryobiology or are doing work in applications relevant to cryobiology. So you could be a biologist, chemist, engineer, or a clinician who has interest in this low temperature biology and medicine. The membership of ICYR is open to any young researchers, which could be anywhere starting from an undergraduate all the way to early career faculty. The requirement is you'll have to become a student or sort of student member of Society for Cryobiology, which is a free membership, and it comes with the perk of being the part of this global community, which we are building. As I said, it's a wing of Society for Cryobiology, and we operate under them in terms of legally and structure-wise. Please feel free to check out webpage and our Discord. We are very active on that. And the leadership represents myself as the current president, Charles, who is in this call, who is the vice president, and other people who are not here, but they are an integral part of. So if you're interested in leading and being the leaders of tomorrow's, please, in December, we'll have the next cycle of elections. So please apply. And a quick timeline. So as I was saying, this ICYR has existed before even a pandemic era, but what happened this 2022, that was the first cryobiology in-person meeting in Dublin after pandemic, and I had a pleasure to attend there. And this idea, talking to someone, Daria, which was a master's student in Iceland working in cryopreservation, we started discussing why don't we use this virtual platform which Cryo is using for ICYR, and that became a reality in 2023 in Minneapolis. We hosted, alongside the in-person event, we have virtual events during the society's annual meeting. Coming to 2024, we had a whole structure, and Charles was on board and other people was in the organization. We had bylaws. We started our year-long event. So instead of just being presented at the society's annual meeting, we started doing every other month kind of events, professional development oriented towards young researchers. And then we started a journal club. In 2025, and we started a professional development award, which was, first of them is sponsored by ATP-Bio's industry, which is BioCoric. So thanks to MPP for it. And then the second one is coming out in fall, should be Expense Bio. So thanks to Brad for it. And then we went outside Society for Cryobiology to bring people into it, and that is Society for Low Temperature Biology in Europe. So just last week, our ICYR booth had a really good turnout there in Poland. Coming in 2026, we will be doing one Young Scientist Conference, which happens with IPCC in Ukraine. This is a virtual conference for any students, and it's free of cost, and the best abstracts would be published in cryobiology, the top 3 of them. So ICYR would be playing an important role, and I have invited SLC as well to join this conference. In future, there is lots of ideas, and some of them, for example, Charles had about bringing a cold award challenge, which would be hosted by Society and ICYR. It would be a huge lump sum amount of bringing researchers from outside cryobiology to solve cryobiology-related problems, and hopefully a summer cryobiology school and course, which can train these young scientists. Our membership has more than 100 people as of now, so which is really grew up from 10 people all the way to 100. Not all of them are active, I'll acknowledge that, but if you see the distribution, we have PhDs as the most, and then we have undergraduate as well as postdoc and other people. One part of this was that most of the Society for Cryobiology annual meeting used to be in a sort of North America focus. And that's where ICYR is trying to build this global community where we have now good amount of members from Europe. And then we are actively expanding beyond, such as Asia Pacific region as well. So just some of highlights as societies. Our liaison is Dr. Rebecca Sandlin. And we in 2024, we in fact hosted along with ATP-Bio DCI, the implicit bias mitigation workshop. So that was a part where ICYR started to come into ATP-Bio. ICYR has hosted a lot of ATP-Bio trainees for our webinars. So you can see, and as I said, this year we had this first professional development award, which really is for any trainees who wants to use that as a supplemental thing to go into a new lab or coming from a new field and wants to explore a cryobiology lab. It could be even an industry, not academic as it's. So not a conference travel award, but something to explore your cryobiology curiosity. In fall, as I said, very soon we'll be inaugurating the Expense Bio Award call. So if you're interested, do apply for it. There will be entrepreneurship event, which will feature multiple cryobiology startups in October/November. And then, as I mentioned, 2026, we'll be hosting the Young Cryobiology retreat or conference with IPCC in Ukraine. So last word is, we are trying to build this international community in cryobiology. So if you're a trainee or a young researcher, this is the place to do, whether it's social event, personal connections, or training in any type of professional skills. And these connections would last for lifelong if you want to stay in cryobiology. Examples of that is, for example, Charles ended up having ATEP exchange to ATP-Bio's lab, even though it was from outside ATP-Bio. So it's a plethora of opportunities in research and connections. So I'll just say, please join and be the leader of cryobiology. Thank you.
Great. Thank you, Lakshya, for that very stimulating talk. And of course, the Scholars Leadership Council, which we'll be hearing from shortly, is a part of ICYR and vice versa. So there's a lot of interplay there. I enjoyed your timeline slide. I'm not sure many on this call will remember. I think maybe Kelvin would remember this, but this was actually started in, I believe, the Pittsburgh meeting in the 90s. And the two people that started ICYR today are probably interesting to the crowd to know. One of them is Jason Acker, who's a vice president at the University of Alberta now. And the other is Gloria Elliott, who is a program officer, program manager at ARPA-H. And she's starting a desiccation program there. So people can go far with ICYR, right?
That is great.
And Lakshya also was part of the SLC, which Alexandra is now leading. But before we jump forward, I just wanted to give everybody a chance to ask Lakshya or Charles, who's also on the call, if they have any questions of the ICYR folks. How did the, maybe you covered it, but maybe you could just say again, how the SLTB, is that happening now, or is it going to happen, or how did that go?
Yeah, so that was last week, 10 to 12 or 13th. It went well. IDP-Bio as part of ethics and public policy. So we presented virtually abstract, and then ICYR, we were able to give all the sort of physical merchandise during the Cryo in Hanover, so which was hosted there. And I see at least, I think, 6 or 7 people registered from that in the intake form. So yes, we are looking forward. And another good part about that SLTB outreach was that we are able to do that without actually going there. So all the European young researchers took the lead from Birgit's lab, and they were representing ICYR as well as the SLC booth there.
Great.
So, right.
Great. And that's just underscoring the international, nature of this group. And SLTB, for those of you that don't know, is the Society for Low Temperature Biology. So there's a long and rich history of cryobiology in the British Isles. And of course, the cryobiology meeting was in Hanover, Germany this last summer. So great. Hearing no questions, we'll move on then to Alexander will tell us a little bit about what's going on with the Scholars Leadership Council. Alex.
All right. Can you all hear me?
Yes.
Okay. Thank you, John. Let me share my screen. Okay. So yeah, hello everyone. My name is Alexander Ulate. I am a third-year PhD in electrical engineering at the University of Minnesota, studying under Dr. Rhonda Franklin. And I'm here today to talk a little bit about some of the advancing collaboration, service, and scholar engagement in the NSF ATP-Bio through the SLC. So I am one of the Student Leadership Council co-chairs, and I will showcase. There we go. All right. So for those who maybe aren't familiar with what the SLC is, the SLC is the Scholar Leadership Council, and the role of ATP-Bio Scholar Leadership Council is to serve as a liaison between the SLC body and the ATP-Bio leadership, voicing any concerns and relaying important information from the trainees to the leadership and vice versa. One of our primary goals is to connect trainees to all of the center resources. This can come in the form of career development sessions, such as our Trainee Tuesdays, information on funding and volunteer opportunities offered by the center, also known as just our trainee meetings with food, any type of formal and informal mentorship within the center, as well as cross-center collaborations. So who is part of the SLC? This is kind of, our trainees is our catch-all term for pretty much anybody who's working as part of ATP-Bio doing the physical research. This could be undergraduate students, graduate students, postdocs, lab technicians, and research assistants. So this year is year 6, and this is the SLC Leadership Council for this year. Along with myself, I have my fellow co-chair, Maxwell, and then we also have our communications lead, Sohail, career development lead, Rasha, and the networking lead, Julio. So the elections for this leadership council is held at every annual meeting in the spring. So the nominations come from fellow trainees. So anyone who's involved in the center, if you know a trainee and you think they would be good for any one of these positions or a leadership role, you can definitely nominate them there. Highly encourage it. And as you can see here, we all have our affiliated universities. So every single center is involved or is encouraged to be involved in the process. So not one university has all the leaders. So it's a very great way of getting a lot of cross collaboration with other universities, as well as giving a great leadership experience opportunity for those who have been part of the SLC Leadership Council. So I'm just gonna give a couple overview highlights of some of the main things that the SLC offers. I know I mentioned a couple of them before, but I'll start off with the Trainee Tuesdays. So these are our bi-monthly meetings that we have for any type of trainee development. This can be from CV and resume plannings, events that we've had in the past, lab demonstrations where we'll invite trainees to set up a camera in their lab and showcase we've done surgeries before. Other times we've had demonstrations on how to use different type of lab equipment, how to set up different type of like chemical setups. So it's a really great way to get trainees involved and kind of see what is everybody else in the center doing. It's a really great way of highlighting all of the different centers and kind of what specialties people all bring to the center. We've also had events on how to set up lab automation for experiments. So it's a great way to kind of get a lot of those research skills that may not be available in your exact lab, but may be available in another lab that's part of ATP-Bio. And then we also have kind of more on the professional side. We've had applying for postdoc, graduate, and professional experiences, which can sometimes include some of the companies that work with us. Maybe they come and give a talk or give a little bit of insight into what's out there for jobs in cryobiology or anything tangential to what ATP-Bio is trying to do. So the next thing I want to highlight is the ATP-Bio Trainee Exchange Program that we offer. So this is a funding that's available to all trainees to kind of take a work exchange within the different centers and member partners. It's a really great way that encourages cross-center collaboration while covering travel and living expenses. So it allows you to pretty much, if you know a lab over on the East Coast has a better machine than maybe what you have on the West Coast, you can take advantage of the ATEP funding to fly on over, spend a couple days in the research lab with different equipment, different people, and then head on back to your center to bring back the knowledge that you gained from there into your center. So it's a great way of meeting other people in the center, kind of growing your network, as they say, to include a lot of other different people, viewpoints, different majors, backgrounds, etc. So I personally have been able to do an ATP-Bio exchange with Massachusetts General Hospital. So I thought I really enjoyed my time and was able to work with groups in working in chemicals that I had never worked with before and didn't have available here in my lab. So it's a really great experience, and I highly encourage any trainees in the center to try and take at least one ATEP program if you can. And especially with last year's ATEP applications, we actually managed to hit our entire funding goal. So it was highly utilized, and you can use it to travel to pretty much any of the ATP-Bio centers, and it's a highly sought after thing. So if you haven't heard about it before, I highly recommend checking it out this coming year. And then we also have our SLC activities that we host at the annual meetings. So when all of the center leaders are gathering together, we have 3 main events that we host for the SLC members. And those are your poster sessions and competitions. So if you want to practice giving posters or presentations, we have that there. The winners from last year's competitions are over on the right. So highly recommend. It's a lot of fun. And then we've also had in the past, if you're into entrepreneurship, they've had the 60-second startup pitch where the trainees are encouraged to come up and present for 60 seconds on their startup idea. And then at the end, a winner is picked out of all of the presentations as the best startup idea. So highly recommend. It's a lot of fun. It's a great way to get some communication skills and practice while working with all of the different centers together at the annual meeting. And lastly, I just want to end with kind of something I've seen grow, especially in the last couple of years as the center itself has grown, is the community outreach part of the SLC. So all of our trainees are encouraged to volunteer at outreach events, be it in the local area or at other ATP-Bio centers. And a lot of the targeting has been towards like K through 12 learning activities. I know Massachusetts General Hospital has done a lot of work on creating different activities about cryopreservation for kids. I've been able to build a heart lab out of just like everyday materials here in Minnesota. And most of these activities are made by the trainees. So if you're a trainee who has experience with outreach, or you have some ideas on how to teach kids about what ATP-Bio does, we're always looking for new activities and ideas. And then we also kind of have, even within the center, trainees can mentor research experience for undergraduates or REUs. So sometimes we'll have the more experienced members be those mentors for new people joining the center. And it's a great way to continue on with the mentorship cycle of you were mentored, now you're mentoring somebody else. And it really helps connect kind of this idea of building the next generations within ATP-Bio as part of ATP-Bio's mission. And with that, that's kind of a general overview of what the SLC does. If you have any questions, feel free to ask. Otherwise, we also have, if you want to get in contact with us, we have an email. But yeah, I think that's all I have. I now open the floor.
Thank you, Alexander. That was great. One thing I'll mention is that this idea of exchange is one that we are growing and not, it's in no way retracting. I think we're putting more and more efforts and resources into it. We see this as a way to not only support the trainees, but it also supports the greater mission of the ERC. And we have exchange both for trainees, which has been well used, and Catherine can say a few words about that, but I think we've used up those funds the last couple of years. But it is also something that both our ATP-Bio partners as well as faculty members can also use to increase exchange amongst our labs and bring in new ideas or take away ideas. So collaboration is really a central theme here, and the exchange programs are really the way we see. It's not that much of a in the context of the whole center budget. It's not that much to actually put in to actually support people to be able to do this, both the trainees as well as industry and faculty. So Catherine, I'm just going to call on you to maybe say a few words about how first what has happened over the last few years, and then also how people can implement or do this.
Yeah, so we are redesigning the process slightly this year, and we are planning on doing 2 rounds. It's one, I think, with a deadline of maybe October for application. And we haven't gotten the documents out, so where we'll select maybe up to 10, and then another round for the last 6 months of the year, where again we'll provide probably 10 ATEPs for trainees. And then we also have the visitor exchange program, which is more for probably people from outside the labs to come visit ATP-Bio labs or vice versa. So there will be more of an internal process that's going to be grouped under ATEPs with more opportunities, and then the VEPs, which are going to be for collaborations from outside to inside ATP-Bio, and with basically 2 application deadlines per year. It's a very simple process. It's just a little form to fill with a little proposal, 2 letters of recommendations, and then we basically, Jennifer, who's also on the call, takes care of all the logistics, and it's been extremely well used, and it has really led to a lot of collaborations and publications between different sites. And yeah, it's a big success story in ATP-Bio, I would say, both for the ATEP exchanges and the other one.
Right.
So if somebody on the call is interested in coming, just contact either Jennifer or myself, and or look on the website, and just we can answer any questions you have.
Thanks, Catherine. Jennifer, did you want to add anything to that?
I guess only a brief mention that Catherine mentioned that there'll be 2 deadlines, and those will be priority deadlines. So we'll review applications then, but if you kind of think of an exchange and it's past that deadline, you can still apply. And if there's still funding available for that sort of semester, we'll kind of take those on a rolling basis. But that was just a way to kind of spread out some of the funds to make sure that there were opportunities throughout the year.
Yeah, that's awesome. And I don't know, since we're literally submitting our renewal proposal tomorrow morning here in Minnesota, we've been going through a lot of our numbers, and maybe it'd be worth highlighting. I'll try to find the page here, but we have some pretty amazing, I think, things to be proud of in our workforce development. And maybe I can just share screen if people would like to take a peek at this. And then Jennifer and Catherine, you can help me interpret any questions here. But can people see my screen?
Yes.
You want me to make it bigger, Lakshya? Let me make it a little bigger. Okay, so can everybody see that all right?
Yeah.
Yeah, so I'm doing this obviously because we also have a little bit of time, but if there are questions, of course, please ask questions of our speakers. But this is 336 trainees in the program, and you can see that, you know, the current enrollment is, I don't know, maybe in a third to a half of that. But we've actually had more than 10 of our trainees go into faculty positions, obviously many more go into industry. And you can also see some of the statistics around our exchange fellowships and RU program, as well as our senior design and engineering research. So maybe, I don't know, Jennifer, if you'd like to say a few words about this, anything more that you'd like to add? Thanks, Emily.
No, not too much other than, yeah, these programs are continuing or evolving. And so kind of keep an eye and ear out for additional opportunities.
Yeah, great. Catherine, was there anything you'd like to add to this?
No, I also wanted to highlight the number of students going to medical school, which is also quite impressive. And the programs in the bottom, the table at the bottom, these are the programs as they're currently, but we're really refining each one of them each year even more. So that's quite it's just the beginning, I would say.
Yeah, and actually, Alexander, maybe you want to say a few words about the 7 leaders per year in the SLC, because I'm not sure. You may have covered it in one of your slides, but you may want to mention it again. It's not just you, right? There's a bunch of other folks.
Yep, we're a whole group. I think there might even be 1 or 2 positions still open. So still looking, if any trainees are looking to get some leadership experience, more than welcome to join.
All right, fantastic. Any questions about any of this that anyone would like to ask?
Catherine, I wanted to ask the visitor exchange one, which you said that is not for trainees though, correct?
Well, ATEP is for trainees, and VEP is, and Catherine, you'll have to remind me, but it's investigator exchange program. Is that what it stands for?
That's how we used to distinguish them until last year. I think this year what we're planning on doing is saying that the ATEPs are for internal exchanges within ATP-Bio labs, and the whether, you know, at any position, any role. And the VEPs would be for external people visiting ATP-Bio labs or vice versa, including trainees. So for example, Charles, who's on the call or was on the call, he came to your lab on a ATEP because he was a trainee. But from next year on, he would be coming on a VAP because he would be coming from outside. So it's just a matter if we've changed roles. We've put all the trainees in one ATAP, in the ATAP grants, and all the people coming from outside or us going outside into the VAP grants.
Yeah, and I think that's kind of what I was going to, like, at times there might be like students, researchers who want to come and who are not at the faculty stage, and we want to make a collaboration. So that would like opening that gives a path.
Well, maybe we could have Charles say a word or two, because we're coming down to the wire here. But Charles, maybe you want to share your experience if you're still on around the ATEP and what the Mensi lab.
Yeah, sorry, I'm on the move a little bit, but I had a great experience traveling to perform some experiments in Dr. Bischoff's lab. Me and Lakshya are working on a cryopreserved red blood cell method where maybe we don't have to wash the cells afterwards. So it's been very exciting, and that was back in February. And me and Lakshya are taking the project further, and we're very excited and grateful for the ATEP program to allow this collaboration. Thank you.
Thanks, Charles.
I wanted to also throw out a comment. ICYR would love to collaborate with SLC more. So we can think of ways to kind of bring the national cryobiology young researcher program and the international one together. I think that would be a huge success. So thank you for letting me speak on that.
Thanks, Charles. And Ken, I see you have a hand up.
Sure. Is this audible?
You are audible.
Okay, good. Very quick question. There are a lot of different ways of going about matching people with locations for exchanges. And the best way is obviously to let doctoral students think hard about what they want to do and what will advance their professional careers. But there's another aspect of this, which is maybe a little more strategic from the perspective of the ERC as a whole. Are there particular areas where cross-fertilization, multidisciplinary and interlaboratory, are identified as being really useful and worth emphasizing? And to what extent are doctoral students being encouraged to be doing the exchanges in a way that might advance those larger goals? So is it bottom-up, student-initiated, or is there an effort to identify priority areas?
Oh, that's a great question. I mean, we could always do more, Ken, to be honest. I think to be more strategic. But for instance, we went to Texas A&M during our annual conference to identify more folks working in food and sustainability.
Right.
And kind of moving into space. And some of those things have come into fruition because of that. And there have been some, I think, exchanges and relationships that have been built. But your point is well taken. I mean, I think if we could be more strategic, that would be great. And Catherine, maybe we can bring that to the CR group.
But it's also a delicate matter because you don't really want to be imposing priorities from above on people that have to be worrying about their careers and can make their own judgments on it. But it's at least worth raising. I also want to mention that some of this goes beyond technical areas of collaboration. Within SynBirk, the ERC in synthetic biology, there were always people in the technical labs that wanted to be working with our policy group. And it was when they were working on something that was exceptionally problematic, weird, potentially triggering public reactions, and they were interested in how policies might be made on it. And so we ended up working with a bunch of grad students from other labs and ended up publishing in weird in places like Nature and Science. I mean, places that were pretty prominent. But just noting that the incentive for technical graduate students to do stuff that would be outside of the technical area was that the prominence, well, a publication in Science is always fun and it's kind of hard to come by.
Yeah, no, well said.
And I'm sorry to be over time. Apologies.
No, it's fine. You can see there's interest in hearing you, Ken. So we're still.
I just wanted to answer Ken about the motivation coming from higher up and being more strategic. that I think, John, has happened quite a bit because we had the seed funds for projects that were actually coming from our, our leadership, which were where were the gaps, the technological gaps that we needed to fill. So we send out like a request for proposals, and it turns out that this request for proposal was very often a kind of a starting point for an exchange between students, like like people would PIs would converge about how they could do this, and then they would exchange their students to through one of these exchange programs. Yeah, and these exchange programs are also open to the other pillars, not just the research pillars, but it just happens that until now we've only had research exchanges, but it's
Okay.
open to
Thanks very
everybody.
Thanks very much.
Well, we're at time. Ellie has asked me to do a quick promo for our October 21st webinar, and that'll be actually at 1 p.m. versus 3 p.m. And Ellie, you have something to show us? It'll be Dr. Terrence Tiersch, our friend from LSU, who also sits on the EP3 with Ken. So this should be a stimulating conversation about community level approaches to safeguard aquatic genetic resources. So with that, I think we'll call it to a close today. I want to thank the speakers and thank you all for attending, and look forward to seeing you all next time. Take care.
Bye.
All right. Bye-bye.